Hey, so welcome everyone. Good morning. My name is Ioannis Panetas, and today I will introduce you to the FlowJo software. So we're going to be talking about basic flow cytometric data analysis using the FlowJo software. I'm an application scientist at FlowJo Informatics, which is part of BDP. You can find my email address here in case you have any questions after the end of this webinar. And please use the Q&A box to ask your questions. And me and my colleague Serena, working for FlowJo, will try to address as many questions as we can here today. So let's get started. So the content of this webinar, at the end of this webinar, we're gonna learn three main functions in FlowJo, yeah? So the very first section is creating groups and keywords. So it's really useful in FlowJo to, when we import our files within FlowJo, to start organizing them in different groups and assigning more information on our samples, which we can introduce as keywords in the FCS files. And this will allow us basically to make our reports faster, not to lose time to, when we have to generate reports of statistics or graph reports, we can make only reports from the given samples that we want to. If it's different donors or stimulation statuses, time points, we can just focus on specific information to generate reports. So that's the first part. And the second part would be the main part in FlowJo, so where we spend most of the time usually, the graphical window. So the graphical window is where we can visualize our data. We can change the axis and see all the different parameters we acquired in the cytometer, and we can start gating. So we can find all those interesting populations we have in our data set and start forming our gating strategy. So I think it's not possible to ask live the question, but you can type it in the Q&A box because I see somebody raised their hand. Yeah, so make sure to use the Q&A box to address your question. Yeah? And in this part, we can also see how we can modify the graphical window. We can transform the axis to allow better visualization and use different tools to share different populations. And finally, the last part will be to generate the report. So this is what we want to achieve. We want to see the comparison between the different samples, and we want to make graphical reports to define the different subsets across the different samples. And we can export those, and then we can do the same exactly for our statistics. So we can create a table of statistics. For example, we can have the MFI, the counts of the cells, and any other frequencies or any other statistics we want to make a table that we can save for our record. Just checking here the chat. Yeah, so it's Serena there also in the chat box. So make sure if you have any question to type it there. Sorry, it appears I made the drawing. Oops. Okay. So for the data set, okay. So for the data set we're gonna use today, we're gonna have some PBMCs. And this was an experiment for identifying, using a phosphoflow intracellular staining, 5 different donors across 4 stimulation conditions. Yeah, so that's our categorical parameters that we want to compare. So here is a schematic of this experiment. So we have the 5 donors. Those are annotated in the FCS file we're going to be seeing soon. as LD1, LD2, etc. So this is coming from living donor 1. So you can, label your samples whichever way you want in your cytometer, but you can also change that name or display name in the FlowJo software. Yeah. So those samples were stimulated first with PMA onomycin, and that's the annotation name of PI in the name of the FCS file, or non-stimulated, which comes from the NS. Yeah. So then we had the surface staining, so for some basic markers, and then a second stimulation step with PMA onomycin, and a control as NS. Yeah. So, and then finally we have an intracellular staining for interferon gamma and phosphor. Files were exported as an FCS, as FCS files from the cytometer. So this stands for flow cytometry standard files, which typically you can export from most cytometers, but FlowJo also supports other kinds of formats if you have, so you can use those and load them into FlowJo and analyze your data. Yeah. So in total, we have here 34 FCS files, which are basically 5 donors and 4 stimulation conditions, which makes 20 fully stained samples. And then we have 14 fluorescent -1 controls to set the gates. So some of the antibodies we use in this panel are difficult. So interferon gamma and phosphor, if you ever try to stain, are quite challenging. So that's why we use fluorescent -1 controls, which is basically a sample, either stimulated or non-stimulated or combination, which lacks one of those antibodies. So it contains the whole panel apart from interferon gamma. And that's an FMO for interferon gamma. And we can use those samples to set correct gates and have confidence that our positive cells are really separated from the negative at a specific point in our plot. So we're going to be seeing how we can use those also to set the gates in the software. Okay, so that's it with the slides. So I don't have much slides. I prefer that we see everything else in the software. So let's get started. Okay, so just check the Q&A box. Okay. Good. So let's open here FlowJo. The latest version you can find is 10.9. So I always suggest that you update and always use the latest version of FlowJo, because we really put work into improving function or integrating more algorithms and tools into FlowJo. So keep updated also always with the latest version. Yeah. So we'll open here a new workspace. Okay. And that's the FlowJo main workspace window. Yeah. So basically what you see is 3 different sections. So typically, like most of the software, we have different tabs here, which will change the so-called ribbon. To get started, the FlowJo tab and workspace tab are just fine. You will find most of the tools there. Then we have the FlowJo button here where you can save the workspace. And then there are some shortcuts here where you can, for example, add samples quickly. You can create groups, create a compensation matrix in FlowJo, the table editor, which we use for generating statistical reports, the layout editor, which we use for graphical reports, and then we have recalculating statistics and importing. You can have access to the BD Research Cloud, which is a new software to design panels or go from acquisition to analysis and support your flow cytometric experiments. Yeah, so this has connectivity directly with this platform of PTRC. In the middle section, we have the group area. So initially, when you start a new workspace, you will have the all sample group and the compensation group. So, and for each group, you will have the size. The size will be basically the number of samples we have in each group and the role of the group that you can assign yourself. Yeah? Whenever we select a group, we will see on the bottom part, the sample area, which samples exactly are within the group. Some statistics like the frequency or any other statistic we choose to display, and the number of cells. Yeah? One of the most important buttons to customize this workspace here for FlowJo, or all the different kinds of windows and functions in FlowJo, is the preferences button. Yeah, so this you will find under the FlowJo tab and preferences. Yeah, so many of your questions can be answered here, like how can I change the display name? Yeah, so this you can find under the workspace. You can use a short file name or the FCS file default name keyword. So you can be flexible in terms of, you know, if you name your samples like tube 1 or tube 2 on the cytometer, you can still change that in FlowJo, yeah, to allow a better and easier analysis. Yeah. Apart from that, there are different tools like changing the fonts and the size of the fonts. You can change the gate. So if you want a specific type of gate to be as your default, the line weight of each gate, annotation position, many different functions that can make your analysis much faster. Yeah. One good example is the cytometers. This is something I like. So especially if you use different kinds of cytometers, you have the option here in this window to save and adjust the scaling from files coming from a specific cytometer. Yeah, so if you use a cytometer with a slightly different scaling, you can find this cytometer on your list here on the left, and you can, for example, adjust the scaling. So if you don't want a default scaling, you can deselect here those options and adjust your minimum and maximum values here. Yeah, and then the next time that you load samples from this cytometer, FlowJo will recognize and adjust the scaling that you wanted in the first place. Yeah, so FlowJo identifies where does an FCS file come from. Yeah, and you can save that and be flexible and not have to adjust that scaling. Yeah, things like that you can have access to the FlowJo preferences. Yeah. Okay, so why don't we get started here? So let's import those PBMC files we were talking about earlier. So here is my folder. So it's called FlowJo dataset. And here you can see the different samples that I have. Yeah, so you see a subset of FMOs, fluorescent -1 control. Besides this, you see the stimulation combination. So we have NS, which stands for non-stimulated. And the second step is PI, which stands for PMA ionomycin. That's both for FMOs and the fully stained samples. The fully stained samples you can see here, LD1, which is the donor one. And this, for example, is a sample of 2 times non-stimulation, so it's a controlled sample. Yeah? And for each donor, we have 4 stimulation conditions. Yeah. So now, how do I import the samples? So either I can drag and drop all of them here and load them into FlowJo. I can also drag the whole folder if I want. You can be very flexible about how you import samples. I typically, that's just my preference. I drag and drop only my FCS files, because when I drag and drop a folder, this will create a new group directly. Yeah. But if I just drag and drop the FCS files, we will see now that I have 34 samples here in my old sample group. And you can see the list of all my samples there. Yeah. So going further, now we can start creating groups, because we might not want to have our FMOs together with our fully stained samples, because the goal of this introduction to FlowJo is to make reports only for our fully stained samples. Yeah, we don't want to have the FMOs together in our graph reports. Yeah, so that's why we will start by creating groups. Let's use one way of creating groups. One easy way is to select the samples you're interested in. So I can select here my LD samples, my fully stained samples, and then I can go under my workspace tab and select group selected samples. Yeah, that's a very easy way. You just multi-select, press group, and then you just type the name of the group. So this will be my donor group. Yeah. Okay, and as easy as that, I made a new group. You see here the donor group with 20 samples within. Yeah? An additional way of creating groups. Yeah. So I see a question here. The question is concerning the preferences tab and cytometer. Are all available cytometer names already visible, or do you have to I guess you want to add them step by step? If this is the case, is there a list of all cytometer names that can be imported manually in the FlowJo software? So Christoph, here the answer is there is no list automatically in FlowJo preferences. So whenever you drag and drop a file into FlowJo, FlowJo will recognize a keyword, which is the $ cytometer, and then it will add it into that list. So if you use FlowJo for the very first time, you won't have any cytometer there. So the more, if I go here, for example, under cytometers, here you see now the list of all the cytometers I have used samples from. Yeah. Otherwise, this list would be then empty. Yeah. Yeah. You're welcome. So let's move on and see another way of creating groups here. So here we might want to have a group for the FMOs. Yeah. The second way of creating group is to press the function here or shortcut create group. Okay. And then this window will appear where you need to assign the name of the group. And this could be my FMO control group. Okay. I will need to save my workspace here because, again, preferences, you can select autosave and have every couple of minutes your workspace saved. So I will set a name which is FMO control. I will set the role which will be control so that it really indicates that that's a control. And then I can use some of the keywords of the files, of the FCS files, to automatically put samples within that group. One of the most useful keywords you need to remember is the $file. So this is basically a keyword which corresponds to the name of your file. So just the file name, which I can use and say to FlowJo that, okay, when I see in the file name that it contains, yeah, so you can use the contain function, and when the file name contains FMO, which is unique for my FMO control, then only those samples will go in that group. Yeah, and that's a very easy way to be flexible, to create groups fast and just assign the samples you want within that group. Yeah, and then I'll just press here, create group. Okay, and you see, as easy as that, only the samples that have in their name FMO, they were transferred in that control group right here. Yeah, and you can make that way as many groups as you would like to have in your workspace. Okay. So within a group, so you can order the samples in a specific way. So here you see those are ordered alphabetically. Yeah, so I'm going from LD1 to LD14. Yeah, if you want, you can double click the column, and this will order them either in an ascending or descending order alphabetically. And you can do that for every single column you have in here. Yeah, so if you want, for example, to sort your samples according to the number of cells, you can press here, double click, and now we will start from the sample with the least cells to the sample with the highest number of cells. Yeah, so remember that you have this flexibility to automatically change the order of the samples. Yeah. Now, the FCS files are basically they stand for flow cytometric standard file, and they basically have a list of keywords that we don't see at this point. But if we want to visualize those, we can press right click on any sample and then press inspect. Yeah, so this will open that main window where on the left you can see all those keywords that were part of this FCS file. Yeah, so you can see, for example, the cytometer. As we mentioned earlier, FlowJo can recognize which cytometer your file came from. And here we have, for example, LSR2. And many different information about the date of when the dataset was acquired, the file name that we discussed earlier, and many more. Yeah, so that's the list of all your keywords. If you want to display one of those keywords, you can press right-click on the columns here and select Edit Columns. Yeah. In that main window, now you can add from the left list to the right list any keyword you want to display. Something very useful is in case you have plate-based essays is to use the well ID. Yeah, so you could use the well ID and add it to the columns here. You could do the same, for example, for the cytometer. You can also do that and add it here as well, date, etc. And after you press OK, so this now will transfer some of the saved keywords that we have in our FCS files into our workspace. Yeah, so now I'm much more. It's much more easy to see. Okay, this sample wasn't that well. I can even sort them out based on the well alphabetically. This is in the first well, second, etc. And I can always be flexible to then press right click again and remove those columns if I don't want them anymore. Yeah. So I can remove here, for example, some of the columns. And then we can, apart from the already saved keywords that we have in our sample, we can also add our own keywords. Yeah. So the way to add keywords, you can find here under the workspace tab and select here add keyword. Yeah. Then FlowJo will ask you for the name of the keyword. And I could ask, for example, here to add a keyword for my donor ID. Yeah, so I can select here donor ID. Okay, and now I created a new keyword. So it doesn't mean that only what keywords we have from the export of the cytometer we will have here in FlowJo. We can add extra keywords ourselves here post acquisition. Yeah, and now I can manually now I could manually input here values for my donor ID keyword. Yeah, so I could, for example, put 1 for my first donor, 2 for my second donor, 3, etc. Yeah. So if you notice that there are patterns of acquisition here, so if you see For example, here I have 4 times the first donor, 4 times the second donor, 4 times the third, etc. So you can use a much more automatic way to fill up this keyword, yeah? And that's possible only if you have a pattern like that. And this you can input here under the workspace tab and create keyword value series, yeah? So here you can select your keyword, which, as we said, was my donor ID. Okay, so this is the keyword I want to fill up automatically. And then you can either have a numerical key or keyword, yeah? So you can choose what fits best for you. And then you can type just the different donors in one column, in the different rows here. So I will add here LD1 for the first donor, LD2 for LD12. So that way I can, just type once all of the values, and then I can set the rules for FlowJo how to fill up that keyword and say, okay, I want 20 values to be filled up. I want to use each value 4 times before moving to the next one. So that way it will make automatically LD1 4 times, LD2 4 times, etc. Yeah, and then I simply press OK, and this will automatically assign my keyword in a much more less time-consuming way. Yeah. Okay, and then we can, after we organize our data this way by adding keywords, different kinds of keywords, create different groups, we can start with our gating strategy. Yeah. So before we do so, just to show you the 3 different symbols here besides its sample. So basically here we have this diamond, which is basically our navigation diamond, which means that whenever you have a window open like that, this is the graphical window, FlowJo just indicates to you that this window is currently open. If you have multiple windows open, you will see that grid here, okay, which means that this window is also open somewhere. For navigation, when you use the graphical window, you can also use those arrows on the top right, and you can go from sample to sample, and you will see that diamond moving also from sample to sample. The second window is basically the quality of your sample. I always suggest to have a look at it, especially if you work with difficult tissues like liver, kidney, or any kind of issues that can affect the way your data were acquired. And double click there, and this will lead you to that window where you basically at the bottom see the median of each parameter across time of acquisition. So if you were like 2 minutes on the cytometer, it will show you here, for example, 120 seconds. Yeah. So what we want here ideally is to have as much stable this green line across the time. Yeah. We don't want to have any different fluctuations or any spikes on the signal, because this could lead to artifacts in our analysis. Yeah, so here we look and we see that everything looks fine. And then you simply need to press OK here. And then your sample will be labeled with a different color, where green is the best color, blue is very good, but not the best. And the problem starts when you have something like purple, red, or yellow, which indicates that something happened during your acquisition. This could be a clog. This could be any type of, hardware issue along the acquisition. If you want to perform this automatically for all your samples, just go to Tools, okay, and then select Check Sample Quality, and this will automatically Yeah, check the quality of all your samples, and it's very fast, and it gives you insights of, okay, I need to pay attention here to see what region I need to exclude from my sample in order not to have any artifacts. Yeah. So I see another question. Unfortunately, need to leave. Get the recording. The recording will be, yeah. So Serena was faster. It's gonna be on the website in few days. So you can go to FlowJo and recorded webinars. Yeah, in 2 days or so, this will be there. You're very welcome. Okay, so then we can start with the gating strategy. Yeah, so we can open one sample, double click. This will open the graphical window. Yeah. So here is where, you know, the main gating strategy will happen. Above, you can see all the different gating tools. So you can use rectangle, squad. I'll try to show you different ways here how you can gate your populations. Here we start with the side scatter versus forward scatter. Okay, so we want to place a gate around here that we think that the lymphocytes would lie. So I can use my elliptical gate, can click here, place a gate around this population. Yeah, and I can place a name here like lymphocytes. I can press OK here, and that way I can, you know, just place a gate here. Yeah. So I see another question. If you have visual examples of when those look bad, I think Serena, I can add a file here. I can do that live. Let me just open quickly a new workspace to show you a bad example. Yeah. So I'll use just one example here. Okay. I'll use this sample. And let's say now you have your sample loaded in FlowJo and you run your sample quality and you see that purple, right? So basically this tells you that you need to pay attention on the sample quality. You open the window here and you see that indeed so we see correlation for most of the parameters here, that at the end of the acquisition, and that's where typically some issues happen at the beginning or the end, we see that we have a spike of those, all those parameters. Yeah. So what you need to do next is not to say, I'm gonna not use that sample at all. What you can do is two things. The one thing I will show you here is that you can use your graphical window, and instead of having you know, the classical plots here, you can adjust your axis to time. Yeah, so remember, your FCS file should have a time parameter, which is the time of your acquisition. Yeah, so that tells you here, just put a parameter. That tells you here that during the first 120 seconds of acquisition, everything went fine because we see we have very homogeneous density of the positive and the negative for CD14. And all of a sudden we had an interruption. So this could be a clog because, you know, the cell stayed for a while there and started clogging. And then we have this interruption that leads to this spike on the CD14 and the rest of the channels. So what you can do here with the basic tools is to place a gate in this area that you before the change point. Yeah, so this is where we have the change point, and I can place a gate and start my analysis now from here. So I can put now my side scatter and forward scatter, and that way I exclude this unstable part. That's the first way I can answer that question. The second way of answering that would be by using algorithms. So we have algorithms in FlowJo that you can use to automatically determine the bad events and the anomalies in your files. And I really suggest that you follow, especially in Europe time zone. We will have Serena, my colleague that is here, will have a webinar on the advanced tools, and she will show those tools how you can automatically and in an unsupervised way filter and exclude anomalies and bad events from your data set. Yeah, I hope this answered your question. Okay, so I'll jump back here to my, yeah, you're very welcome. So let's start now with the main part of the analysis. So we have here our lymphocytes. So now I can change my gates here. I can add, I can change all parameters, add forward area versus height, and I can place now a polygon gate, okay, around my single cells. Yeah, so I can add here single cells. Remember, those gates can be changed, right? You can either adjust the whole gate or adjust the points of that gate. Yeah? And then I can move forward with my gating. Okay, all this gating strategy will be reflected under my samples here. So you see now I have lymphocytes, single cells, etc. Yeah? Let's go further. I can now go from single cells to a live dead staining. So now you see I have two populations here. I need to focus on my negative because those are my live cells and the dead cells are somewhere here. Yeah. The current visualization we have is pseudo color, which means high density of events. We have a red color, low density blue color. It's like heat map. So if you want to visualize differently the population, you can go under the options and use, instead of a pseudo color, you can use, for example, first of all, you can do the pseudo color more smooth. So that reveals much better the two different populations, and you can also show the outliers. You can also use counterplot, which is also a very classic way of visualizing your data, where instead of this heat map, you have those counter lines that show you the density of your populations. Yeah, so you can combine this with outliers, and you can also have different levels of countering. Yeah, so you can have more or less lines to reveal the different populations. Aside from those, we also have the typical dot plot, or in that case, since it's a live dead staining, I could also use a histogram. Yeah, I could use a histogram and place a range gate here on my alive cells. Yeah, so I can set here live. Okay, and I can adjust this gate as well. I can adjust the points and the range gate here. So then I can move on from here. Of course, you can also change the colors here. So you can change, for example, if you want, you know, your dots or your histograms to have a different color, you can adjust that over here. So let's move forward. I can go now to my live cells and select my HLA-DR versus CD3. Okay, so in that case, so now we have two fluorescent parameters. We would want to gate on our CD3 positive cells. Yeah, so in that case, I can set maybe just a rectangular gate to show you how it works. I can place the gate here and name those CD3s. Yeah. Okay. Another nice way to visualize your data So is the color map axis. Yeah, the color map axis allows you to have a third parameter, which you choose yourself from that list here, to be displayed as a heat map. Yeah. So if I select this color map, so I'm just trying to follow up with the question, drag and drop the gate on the next sample or apply the gate. Yes, so this I will show soon, because now we're focusing on one sample. So I use one sample to do all my analysis, but then we will apply this to the whole group to see how it works, and we can make adjustments from there. Yeah. The color map axis. So I can select this third parameter. So for example, I can add here my CD4 T cells, my CD4 parameter. And then I can see that in this area I have, maybe I can hide a bit this one. So you see I have high CD4, yeah, fluorescence in this region versus the rest, which is green. Okay, so according to this scale here, and the scaling of your parameter, you will have different colors according. So that's a nice way to see what you gated. Yeah, so now I know, okay, my interesting population that I want to study next is lying here, so I can place my gate round here. Yeah, and then I can go down from here and separate between CD4s and CD8s. Yeah. So there are some cases that you might import samples in FlowJo and you see that some of your events are on the axis, or that you have a lot of white space between, here, for example, you can see for CD4, all this white space maybe is not necessary to have, right? Because there are no events here. Remember, you can always transform the axis, yeah? If this you can do by the transformation button here, the T button, you can select customize axis. And here is where you need to adjust those histograms to have as much separation as possible, and also to exclude as much white space as possible. Yeah. So typically we use by exponential display here for flow parameters, because it displays much better those near negative values. And here we can adjust our axis. So I can, for example, here add more decades here if I wanted. And if I want to trim that white space here, I can adjust the width basis here, that region. So I can decrease that. And you see now that I eliminate a bit of the white space. We're not changing anything on our data, right? We just change the way we displayed it in the window. You can do that for one or multiple parameters simultaneously if they have the same scaling. And in addition, you can press, aside from apply, you can also press save. So save, remember, this will save that scaling on the cytometer preferences. And that means that the next time that I add a sample from the using this channel from that cytometer, that exact scaling will be applied to my new sample. Yeah, so remember that button, it's important because it can really save you time of adjusting the scaling of different parameters. Yeah. Okay, press apply. So you see I trimmed a little bit of this white space here, and now my my scaling is much more appropriate for this data. Okay, let's set now a gate for the CD8 population. I will use the pencil gate. Okay, pencil gate, you just draw your gate around. If you have a very steady hand, this is a great tool. Okay, you can select here your CD8s, and then you can adjust it as well if needed. Yeah. And then I will go back. Okay, so this is the level we wanted to reach here. Okay, And now, going back to the question that was asked before, which is, okay, we have the gates for the one sample only, yeah? How can we now apply these gates to the rest of the samples within that group? I'll show you how there are two ways. The easiest within the same group is to multi-select all of your gates, like here, and press then right-click. Press here right-click and copy analysis to group. Yeah? Copy analysis to group, and that will apply all of our gates in all of the samples. So those are the exact same gates. And now you can see your gating strategy under your group, which means this is a grouped on gating strategy, and I cannot make now, I cannot delete any gates from the sample area, but only from the group area. Okay. If you want to make adjustments, you can go here to the to any of the gates. And now that you have the same gating strategy, you can use those arrows to navigate from sample to sample. Yeah, so you see here, I go from sample to sample and check how my CD8 T cells look like. Yeah. If I need any adjustments, I can do that. So I could, if I need to expand that gate, I can do that, like here. And this you will see, it will indicate that this gate now is not the same as the rest. So this is indicated by the black color instead of blue. And in that case, I can press here right-click and copy analysis to group. So now I applied this gate, this new gate, to all of my samples. Okay. Okay, so now we have our gating strategy. So applied to the group, but what you can also do is drag and drop gates from one group to another. And here it would be important for the gates of interferon gamma and for phosphor. Okay, so the way you can do that is select your gates from that donor group and drag and drop them into the group here. So there's no shortcut for this, but you can drag and drop those gates into the FMO control group. So now I have exactly the same gates here. Yeah. So, and now I can use my FMO control. For example, I can go to my interferon gamma. So you see I go to my CD8 population here, and I will select interferon gamma. Okay, so now obviously in this FMO control, there was no interferon gamma antibody, which means now I can set a gate, okay, somewhere around there for my interferon gamma subset. So that's an FMO derived gate, yeah, that I can use then on my fully stained samples. So how can I do that? I can simply take that gate from here that I made on my FMO and drag and drop it onto my CD8s on the donor group. Yeah. In addition, I can do the same for my phosphor. I go to my CD8s, put here phosphor on my axis. Here I have only few outliers. Okay, I can place a gate around here, phosphor. Okay, press OK. And then I have a phosphor FMO derived gate, which I can apply back to my CD8s. Okay, that's the classic methodology we use for applying an FMO control gate into our samples. Yeah, and of course, then I need to double check, right? I need to select my CD8s here, put interferon gamma on my axis, and then I can see my gates. Yeah, this is an FMO gate. I can go from sample to sample and see, okay, that gate is really spot on using an FMO control. So here I could also get without the FMO, but I'll just show you the best practices of how you can use an FMO. Yeah, and I can do the same for my phosphor. Okay. Okay, so here we see some of the gates are not, I think I saw one population. Oh, no, they're mostly all right. Maybe I should expand this according to the FMO. So I might need to go back and forth to, again, to apply the gate, yeah, according to the control. I did it fast here. One way of, if you don't want to every time apply, make a change here and then apply to the rest of the group, what you can actually do as a tip is you can select, right-click on your group, go to Edit Group Properties, and select the option synchronized. Yeah, so this option synchronized, whenever you change a gate in one sample, it will change it automatically for the rest of the group. I really suggest this option if you have already established your gating strategy and you just want to, make tiny adjustments to some gates. Yeah, and then I can press apply changes. Okay, and now you will see, as an example here, I can put my interferon gamma or my phosphor better. Okay, so you see now if I change that gate and I put it here, this will be automatically put on my next sample. Yeah, so any adjustment I make automatically applied to the rest of my samples. Yeah. Okay, so now I'm ready with the analysis. So now I can move forward to my reports. Yeah, so let's use the layout editor now to make some reports. The layout editor you'll find at the top as a shortcut. So it's this ruler, L, any way you visualize it. And here, basically, we can add a new layout, name it graphs. Okay, this is where we're gonna make our graphs. Can assign it a color, and the way it works is with pages. Yeah, so if you zoom out, you will see I have an infinite number of pages here, and I can adjust the size of the page from the bottom right corner. Yeah, so I can choose, how many plots I want to include in here, and one way to add samples in here is by pressing, by drag and dropping this sample, any population in that area. So I can drag and drop my CDH here. Okay, so I can zoom back in, of course, because I went too zoomed out. Okay, something like that. Okay, and now I have my CD8 population here. If I want, I can double click here and I can change still the axis. So instead of CD8, I would want to have, for example, interferon gamma. I can add smoothing. I can use large dots, change the scaling, the annotation. If you want to exclude the gates or not show the axis, you can do that as well. You can add histograms on the Y and X axis, change the fonts if you want larger fonts, and many, many different modifications you can do. Yeah. After you make the modification, you press OK, and now you get here your plot as you wanted it. Yeah, for interferon gamma. Yeah. In addition, you can the easiest way to reproduce this plot for another parameter is to press right-click, and then you can add, for example, you can press duplicate. Okay, and then you have the same exact plot, where now instead of interferon gamma on the x-axis, I can double-click and select under the specified tab, I can select phosphor. Yeah, so because we wanted to have those two together. Yeah, so now I have phosphor and interferon gamma for the CD8s. Yeah. In addition, you have other options, like you can add for example, ancestry, which basically will show you all the gating strategy of your that you followed in this for this sample to reach the interferon gamma positive cells. And honestly, there's tons of options. You can go ahead and explore all of them and see what fits best your data. Yeah? Okay, and once you're ready to make a report, so there's another question. Can you adjust your gates directly in the layout editor? No, not directly, but what you can do is to press right click on the plot and select open original graph. That's the only way. And here you can also adjust the gate, and this of course would be reflected in the layout editor as well. Yeah. There's no direct way, but there is a shortcut to reach the graph window directly. Okay, so once you're ready, I typically adjust the size of the page again to fit my plots, and then you need to make the so-called batch report. Yeah, so we need to press here, create batch report, to repeat those plots for all the samples within that group. Typically, I select here the number of columns or rows that we're gonna use. So I'll use 4 columns, which will make 4 pages for one donor. And then I will select separate pages. So that's a trick that you can avoid having any plots, in regions like in the middle of the page. And then you need to adjust again the page. So remember to press separate pages. And then it's very easy because everything would be separate. Yeah. And once you're ready, you can make a new layout where you can either print it, make a PowerPoint, PDF, any kind of format that you would want to have. Yeah. So I can press here, for example, create batch report. Oh, sorry, I moved my file. I can go back here. Apologies. Okay, so you can be flexible and then go back. If you missed a plot or if you didn't have the format you wanted, you can always delete, okay, the layout, and then you can, you can make again a new layout here. Yeah, so you can do as many as you want. Yeah. So separate pages. Okay, and now you see I have 20 different pages from my samples, going from the donor 1 to the different stimulation conditions, to the different donors. Now we have the second donor, and I can export still that as a PDF, SVG, and TIFF file. So SVG is also much more flexible if you want to use with Illustrator. If you want to make publication graphs, we have the best formats to use as SVG, TIFF, and PDF. Yeah. And then, of course, you can make any kind of overlays as well. So I see here there is a question. Okay, Serena addressed the question with the FlowJo website. So we have different ways of overlays. So basically that means you can drag a sample or a population, like the CD8 from the first stimulation condition. And then on top of that, you can add the second sample. Yeah, so this will give a different color for a different sample. Yeah, so then I can add the third one, fourth one, as many as I want. But remember that those now are, on top of each other, and you cannot visualize them really well. But what you could do is you can use histogram. So you can press double click, select histogram, okay, press OK. And then you can even, if you want, you can add a different kind of overlay, not overlaid, but offset. Yeah, so that would mean I can have offset where we don't have any overlay, and I can easily see, you know, my non-stimulated versus the stimulated samples. I can also have a half offset that was mentioned also here. Okay, there's a nice link if you want in the Q&A box about how to make those overlays and how you can customize your gates and everything. And you know, that's the best we can have, you know, one donor for all the stimulation condition to show phosphoric or interferon gamma. Yeah, that's the most informative plot we can have right here. Okay, that's with the graphical window and layout editor. And what if we now want to make some statistics? So to make statistics, we will use one of our populations. So we do have some statistics like the frequency and the number of cells that you can see right here, but we can add more dedicated, more specific statistics. So you can just press right click on your population and then select add statistic. Yeah, so what all this list of statistics you can add if you want. For example, I could use median, geometric mean, CV, standard deviation, all is here. Then you select your population, which is already selected as CD8, and your parameters. So I will select interferon gamma and phosphorus, and that's exactly what I want to study. Pressing then add, we'll add those statistics under the CD8. So it's you know, this Greek sigma letter, which shows you that you have some statistics under your CD8s. So now those, of course, we're not going to write down manually one by one, but we can make a table out of it. The table we will create on our table editor. So the table editor, similar to the layout editor, is not for graphs, but for statistics. So you can find it here on the shortcut, pressing table editor. And here we drag and drop our statistics. Okay, I can drag and drop those statistics in the table editor. And here you see a preview. So the table is not ready yet. We have a preview of the different statistics that we will get. And I can add even more information. That's very nice if you have keywords or well ID or any kind that you want to visualize. You can add them under the Edit tab. For example, I can add here keywords like my donor ID or my stimulation ID. I will add here my donor ID, and maybe I could even add my well ID if I wanted, or anything from the metadata of the FCS files. Apart from this, you can also select different kinds of visualization. So you can select your statistics. You can add a heat map. That's nice to visualize quickly the differences in a heat map, low versus high values. And once you're ready, you simply press here, create table. Okay, and that will create that table with all of our statistics and our keywords. You can sort it out based on how you want. And then we can see geometric mean of phosphoric, how it goes from the non-stimulated to the stimulated goes high, and that repeats from donor to donor. And in our case, we could see, for example, that the highest response of phosphoric we have for donor number 4. Yeah, so we had, after stimulation, we had 610 of geometric mean of phosphoric, and similar pattern we see with interferon gamma, but on a different donor. This can be exported. I can press File, Save As. You can save as Excel, CSV, any kind of format that would allow you then to do your tests, statistical tests, and comparisons. Yeah. Okay, and That's with the table editor as well. And finally, I'll show you how to save the workspace before we wrap this webinar up. In the FlowJo tab, you can select Save as Workspace. So workspace will basically create this file you see on the top right, which always need access to your FCS files. So you need to have, together with your workspace, FCS files that FlowJo need to reach and calculate. Yeah. If you want to have a zip file or a file that contains both the FCS files and the workspace, you can save as an archive file. So archive file is larger, obviously, because it has the FCS files and the workspace. But at least you don't need every time the files to be shared, each time you share a workspace. And finally, we have the template, which will basically save everything what we have done here, the gates, the layouts, the statistics, and everything, but without samples. And that way, when you have new samples of the same experiment, you can load those into FlowJo, and then it will recalculate directly all those. Of course, you might need some adjustment on the gates if you have any batch effects from time point to time point, but it will really save you time not to do exactly the same analysis from scratch. Yeah. So with this, I would like to thank you all very much for joining, and thank you, Serena, for answering all those questions, because I had to speed it up. So thank you very much, and see you very soon in another webinar. Thank you.